Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to our fourth lesson on genetics. Last time, we learned about the practical applications of crossbreeding. In today's lesson, we will learn about the different stages of meiotic division. We will also describe the significance of meiotic division and crossing over. If you are ready, then we will get started. Let us begin by defining the term meiosis. Meiosis is a special type of cell division that is necessary for sexual reproduction. In animals, meiosis produces gametes. Gametes are sex cells, like sperm and eggs. In other organisms, like fungi, meiosis generates spores. Meiosis is divided into two primary stages, called meiosis I and meiosis II. Each of the primary stages is divided into several sub-stages, so we have a great deal to learn. This is a diagram that shows us the stages of meiosis I. As you can see, the four stages are called prophase I, metaphase I, anaphase I, and telophase and cytokinesis. Let us study the details of each stage, one at a time. During prophase I, a segment of DNA is exchanged between homologous chromosomes. This process is called homologous recombination and frequently results in a chromosomal crossover, which is an exchange of genetic information. The newly formed combinations of DNA that are created are a major source of genetic variation and can even result in beneficial new combinations of alleles. The paired and replicated chromosomes are called bivalents or tetrads, which have 4 chromatids. Metaphase I describes the process of homologous pairs aligning along an equatorial plane that bisects the spindle. The chromosomes are independently sorted because of the random orientation of each bivalent with respect to each other. In anaphase I, bipolar spindles called kinetochore sever the recombination nodules and pull the homologous pairs apart. Since each chromosome has only one functional unit of a pair of kinetochores, whole chromosomes are pulled toward opposing poles, forming 2 haploid sets. Each chromosome still contains a pair of sister chromatids, and prepare to divide down the middle. The last stage is called telophase 1. When the chromosomes arrive at the poles, the division is effectively over. Each daughter cell has half the number of chromosomes, but each chromosome consists of a pair of chromatids. The spindle network disappears. A new nuclear membrane surrounds each haploid set, and the chromosomes uncoil. The pinching of the cell membrane in animal cells is called cytokinesis. This also describes formation of the cell wall in plant cells. When this occurs, the two daughter cells are finally created. That is quite a process. Let us pause for our first activity. In a moment, you will see the initial stage of meiosis called interphase, along with the 4 stages that we have learned about. The stages will not be labeled, nor will they be in the right order. Working with a partner, do your best to copy each stage, put them in the correct order, and label them appropriately.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Have you finished? If you worked quickly and well, your result should look something like this. If it does, great job. Meiosis 1 may seem like a complicated process, but it is vital to all life. We must do our best to understand it. Now we must learn about the second primary stage, Meiosis 2. It is the second part of the meiotic process, and the end result is the production of 4 haploid cells from the 2 haploid cells that resulted from Meiosis 1. Fortunately, the names of the stages of Meiosis 2 will be easy for you to remember. They are prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, the nuclear envelope disappears along with the nucleoli. The chromatids become shorter and thicker, and the centrioles move to the polar regions and begin to arrange spindle fibers. This is followed by metaphase 2. The centromeres contain 2 kinetochores that attach to the spindle fibers at each pole. Notice that the cells are rotated 90 degrees from their relative positions during Meiosis 1. In anaphase 2, the centromeres are cleaved. This lets microtubules that are attached to the kinetochores to pull the sister chromatids apart. The final process, telophase 2, is marked by the uncoiling and lengthening of the chromosomes. As in telophase 1, the spindles disappear, nuclear envelopes form, and cell walls form to produce 4 daughter cells, each with its own haploid set of chromosomes. Are you ready for our next activity? Once again, the 4 stages we have just studied will appear on the screen. Please do your best to copy each one in the correct order and label it appropriately.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello again, students. Here is the proper representation of the stages of meiosis II. In order, they are prophase II, metaphase II, anaphase II, and telophase II. I am sure you did not have too much trouble with this activity, given the practice that we had with the stages of meiosis I. Great work, everyone. Today, we have learned the stages of meiosis I and meiosis II. We have also learned how chromosomal crossover results in new genetic sequences. Until next lesson, thank you, teacher. Thank you, students.